We're going to continue with the lecture on drugs used for diabetes, and in this part of the lecture we will see several different mechanisms of action for diabetic drugs, and the first of these is Acarbose, which is actually a natural product as discovered by high throughput screening. And over here we see the structure of starch, which is a three-dimensional polymer made up of glucose units. And if you look more closely at the structure, these glucose units are joined together by 1,4 glycosidic linkages, as shown here and also here, where it's carbon 1 linked to carbon 4 of the next glucose units. And that gives you a linear polymer, which is then cross-linked to give this three-dimensional network by 1,6 glycosidic linkages between these long chains. And in the 1,6 linkage, as shown here, it's carbon 1 of one glucose linked to carbon 6 of the next glucose. And this is the structure of starch, so it's a suitable storage vehicle for glucose. And this then needs to be broken down back into glucose in order to provide energy. And this is done in humans by these two hydrolyzing enzymes, the amylase and glucosidase, which break down these glycosidic linkages present within starch and produce glucose. And if this process can be inhibited, that reduces the absorption of glucose from starch in the diet, and that is beneficial in the treatment of diabetes. The hydrolysis of these glycosides by the enzymes involve this transition state shown here. So if you imagine this is one glucose linkage linked to the next glucose linkage, the hydrolysis is carried out by water, and the enzyme is accelerating this reaction using basic and acidic residues within the active site of the enzyme. So that gives you an activated version of water, which acts as the incoming nucleophile. So this bond is starting to be made as this bond is starting to be broken. And in the transition state for the reaction, you can see due to that there's this partial bond being formed, another one being broken. And furthermore, the loss of this leaving glycoside is promoted by the lone pairs on the adjacent oxygen, which can help stabilize the developing positive charge on this carbon. So that's why you see this partial positive charge character. And also as a result of this stabilization, this carbon here is flattening out to a more sp2 hybridized state compared to the sp3 that it started with. Once the reaction is complete, then we have broken this glycosidic bond and given the one unit and the other. And this being the mechanism of the enzymatic reaction, if there are molecules that resemble starch, they might be recognized by the enzyme as well. Even better if they recognize the transition state of the enzyme. Remember that enzymes work by accelerating chemical reactions, and the way they do that is by resembling the transition state so that molecules that look like the transition state have a high affinity and are stabilized by the enzyme as well. So if you have a molecule that looks like this transition state, then it will hopefully be recognized by the enzyme and act as a competitive inhibitor. And here are three examples shown here. And the one that's clinically used in the UK is Acarbose. In addition, there's Miglitol and Voglibose. And what you'll see in all three of these molecules is they do have a carbohydrate-like structure. So they kind of look like glucose. So they have similar hydrogen bond acceptors and donors positioned in the same way as the starch substrate, and thereby can be recognized by the enzyme. They have additional features that aid in recognition. So you'll see a nitrogen here, and also in Acarbose here, that under physiological conditions will be protonated and bear a positive charge. And that can also aid in enzyme recognition because in the transition state, this oxygen has a partial positive charge. And we'll look in more detail in Acarbose on the next slide. And here you can see the structure of the starch itself. So here's the monomers of glucose, and this is the glycosidic bond that's being broken. And part of that is similar in the Acarbose inhibitor, and you can again see glycosidic bonds. But when you come to this final unit, you see that glycosidic bond has a nitrogen, so it's a CN bond, and also there is no oxygen within the ring as you would normally see in a carbohydrate. So this bond is not a glycosidic bond, so it's stable and it's not cleaved by the enzyme. And that is important because that means the drug will not be destroyed rapidly by the enzyme that it's trying to inhibit. And to aid enzyme recognition, you can see features within Acarbose that allow it to bind with a high affinity because it looks like the transition state of the hydrolysis. And we have the nitrogen here that will be protonated, so that aids in enzyme affinity. And as I mentioned in the previous slide, in the transition state, the six-membered ring is flattened out due to this sp2 hybridization. And that is what you see in Acarbose. There is an alkene here that gives it that sp2 hybridization. And the rest of the molecule, these OH groups, are in similar positions as you'd expect in glucose. So all these features allow Acarbose to bind with a high affinity to the enzyme and be a competitive inhibitor, as is shown here. So the enzyme alpha glucosidase is sitting in the GI tract. So if you look at a blow up of the surface, you will see these small tiny projections, the microvilli. And if you look at them in even higher magnification, this is where the enzyme alpha glucosidase is present, and it would normally recognize an oligosaccharide from starch or smaller fragments, bind to these, and start cutting the glycosidic bond, and that will give you the glucose that is absorbed into the cell. When Acarbose binds, it is recognized by the enzyme, but it's not cleaved at the stable bond, so it can sit there and act as a competitive inhibitor that prevents glucose from binding. And by doing so, that's then preventing the uptake of glucose from the metabolized starch. So Acarbose is a competitive inhibitor of the alpha glucosidase. And in terms of its physicochemical properties, you can see it's a rather large molecule. It also has, due to all these OH groups present, it's a very polar molecule, so it has a very negative log P. And in similar way, it has lots of hydrogen bond donors and acceptors. So these are all features that would predict that this molecule is not orally bioavailable. However, it is used orally, and the reason it is successful is that all these guidelines are intended to predict passive absorption through a cell wall so that the drug can be absorbed in the GI tract and then get into systemic circulation. However, in this case, this drug does not need to be absorbed because the target is actually sitting there in the cell membrane of the intestinal tract. And this is a drug that does not need to be absorbed and does not need to be transported to other parts of the body. So it's right there where it needs to be used. And for that reason, these guidelines don't really apply to it. And this was a drug, as I mentioned in the beginning, which was discovered by high throughput screening by Bayer about 40 years ago. And it's actually produced by microbial fermentation from a soil microorganism. And it's a relatively safe drug. The main side effects are gastrointestinal. And these are due to the fact that when the drug is taken, the starch is not being digested fully and taken up as glucose. And the starch that remains and the saccharides that remain can then be used as food by the organisms that live within the gut, and this then leads to side effects like diarrhea and flatulence. And this is a drug that's typically used when other more common antidiabetics like the metformin and the sulfonylureas that we covered in part 1 are not tolerated, or for some reason they're contraindicated. And it's also a drug that is better tolerated in the Oriental population. And finally, this is a drug that works in the GI tract by reducing the absorption of glucose from dietary starch. So this is a mechanism that is independent of insulin production, and for that reason this would be a drug that can be used if the patient does not have functioning pancreatic beta cells. The next class of drugs, of which there's one example that's clinically approved here, are the PPAR agonists, and this is another example of drugs that were discovered more or less through serendipity and without knowing rationally why these compounds had a particular effect. And this is again goes back to a long time ago. There were these simple carboxylic acids that were reported to have an effect on lipid levels in humans. And eventually this led to additional compounds in this series like chlorfibrate, which was at one time approved for hyperlipidemia. And although the mechanism was unknown, pharmacologically, long-term treatment with these compounds led to proliferation of the peroxisomes which is a cellular organelle, and this later was used in the naming of these compounds, as we'll see in the next slide. And these compounds were worked on by various pharmaceutical companies. This is one of the early ones that was used as an anti-diabetic agent, and it showed promise, and the mechanism of action was only discovered much later, and that was revealed to be the fact that these compounds are acting as agonists on a family of receptors, and these are the peroxisome proliferator activated receptors, or PPARs. So this is based on the fact that these receptors get activated and then lead to the proliferation of peroxisomes. And these PPAR receptors are examples of the nuclear superfamily of transcription factors. So these are receptors that bind to their ligand, and then they are within the cell nucleus, and each receptor has its own preferred sequence that it binds to in DNA, and by doing so then it can regulate transcription at that site. And as shown on this slide, there are endogenous ligands for these receptors, which can be fatty acids consumed in the diet. It can also be metabolites from fatty acids such as prostaglandins or leukotrienes. And once bound to their receptor, and usually these receptors are then bind as a heterodimer with the retinoic acid receptor, the RXR. So in this heterodimer, PPAR plus RXR, they then behave as shown in this expanded cartoon here. They bind as a heterodimer to DNA at particular sequences, and this will then lead to transcription. And the genes that are downstream of these elements are involved in various important diseases, and activation of this receptor has a beneficial effect in terms of the treatment of diabetes. And an example of an endogenous ligand is shown here, and we'll see more on the next slide. It's a fatty acid, and the drug has the same effect of binding to the receptor and acting as an agonist. And within the PPAR family, there are different isoforms of these enzymes. So there's PPAR alpha, beta, gamma, and delta. And the delta is particularly associated with diabetes. And these receptors can be bound by various fatty acids, as shown here. And now there are also synthetic drugs that will also bind with high affinity to these receptors. And in the case of the endogenous ligands, if you look at a typical fatty acid, you have this one functional group, which is the carboxylic acid, which will be deprotonated at physiological pH. And then we have a long tail, which is very lipophilic. And in the drugs, the carboxylic acid is simulated by a different functional group, usually this thiazolidine dione. And in this thiazolidine dione, this NH proton is next to two carbonyl groups, so which are withdrawing electron density, and it's a very acidic ionizable proton. So this NH here within this 5-membered ring is a mimic of a carboxylic acid and designed to be ionized. In the rest of the molecule with the endogenous ligand, we see a lipophilic fatty acid, which is not ideal for a drug in terms of bioavailability. And what you'll see in the drug molecules in this flask is that they do have a certain lipophilicity to them and a certain length, which is similar to what you see in the fatty acid. And in addition, you'll have polar components there that will help reduce the lipophilicity. So in pioglitazone, you see a nitrogen here and an oxygen here. And there are many compounds within this class, some preclinical and some that have entered clinical trials and some that were approved. At the moment, the only one that's in use in the UK is pioglitazone, which is shown here. And you can see the different examples within this class have the similar properties. They have this thiazolidine dione, which is the ionizable portion, and then some lipophilic component, and then a tail where there's something else added, often with a polar component for pharmacokinetic reasons. And in terms of the physical chemical properties, these are usually small molecules that would fit guidelines for oral bioavailability as shown here, and they are administered orally. And this one compound that's approved is pharmacologically similar to others in this class, but it has an improved safety profile, whereas another example within this family, rosiglitazone, was withdrawn due to the cardiovascular side effects. And these PPAR agonists can be used in combination with other anti-diabetic drugs. Their effect is more at the level of cellular metabolism, and it's independent of insulin secretion. So these can be used in patients whether or not they have functional beta cells. And it's not a drug that causes a side effect or hyperglycemia. On the other hand, it's associated with weight gain and fluid retention. The final class of anti-diabetic agents that we'll look at in this part are inhibitors of a cotransporter. These are in fact the most recent and newest class of anti-diabetic drugs and appear to be quite promising agents. And these are a family of drugs that were discovered thanks to inspiration from natural products. And it was known for some time that there are compounds in the apple tree bark that had this pharmacological activity that was potentially beneficial in diabetic patients, and they had been used for that purpose, even though the active principle was not known or the mechanism in the early days. And later on, the pharmacologically active compound was shown to be this natural product, fluorescein, which is a flavonoid. This is the flavonoid part of the molecule, and also a glycoside. Here's the attachment to glucose. And it was more than 50 years after that that the actual target of this compound was elucidated, and it turns out to be in the kidney. And within the kidney, which is the major site for excretion in the body, we have these tubules, and if we expand up the magnification of this tubule, you can see a kind of a proximal and a distal area of the tubule. And within the kidney, there are transporters whose function is to reabsorb all of the glucose. So we consume a lot of glucose and we produce glucose, and some of it will be in the urine ready to be excreted. However, this would be wasteful from the body's point of view. So normally any urine that contains glucose will then be filtered so that all the glucose goes back into the blood circulation. And in normal humans like ourselves, we filter about nearly 200 grams of glucose a day by this mechanism. The reabsorption is carried out by these two co-transporters, as shown here, SGLT2 and SGLT1. Firstly, there is in the early part of the kidney tubule, there is SGLT2, which has a very large capacity, even though its affinity for glucose is relatively low. And because of its large capacity, this reabsorbs about 90% of the glucose that makes it to the kidney. And this is a co-transporter where it's transporting a molecule of sodium ion and glucose at the same time. And so it's an associated process where both get through, and this then leads to the glucose then being reabsorbed into the bloodstream. Any glucose that is not scavenged by SGLT2 will make it further down into the tubule, and there it will be captured by the next co-transporter, which is SGLT1. And SGLT1 is a low capacity transporter because most of it has already been removed by SGLT2, but this will then remove the remaining 10% of the glucose. And in normal humans, as I said, this works very efficiently, so we normally don't produce any glucose in our excreted urine. And in diabetics, often the system is aberrant, and you get a higher reabsorption in diabetics. And these are people who are already having high sugar levels, and then they are also having even higher than normal reabsorption of glucose, which contributes to that higher sugar levels. So if you can interfere with this process, then that should reduce the amount of glucose reabsorption from the excreted liquid. Now, the natural product in apples, the florizine itself, is not very suitable for use as a drug. Firstly, there are two co-transporters, SGLT1 and SGLT2. And SGLT2 is the main kidney co-transporter, low affinity but very large capacity. Meanwhile, SGLT1 is widely expressed, and it's also present in the intestine. So ideally, we don't want to inhibit SGLT1 because then it will lead to side effects in other parts of the body, and we'd like a selective inhibitor of SGLT2. And for that reason, florizine, from a pharmacological point of view, is not very suitable for use. But there's an added issue with the natural product, and that is that this glycosidic linkage is relatively unstable in vivo and rapidly broken down by glucosidase enzymes in the same way that starch is broken down by them. And when the molecule is hydrolyzed, the flavonoid part of the molecule that's produced upon breakdown is actually toxic. And there were various pharmaceutical companies that tried to come up with better versions of the natural product, and one of them is shown here. So there's been changes to the part over here, the flavonoid part, to make it less toxic. And here's the glucose part. And this had moderate activity and made it into phase 2 clinical trials before it was being discontinued. These are other examples down here of other clinical candidates. And these were compounds that appeared promising in animal models, but once they went into human clinical trials, the activity was less prominent. And all of these compounds have a glycosidic linkage, just like the florizine. And for that reason, they will still be susceptible to hydrolysis by glucosidase. And the fact that they failed in humans but worked quite well in these animal models suggests that humans have a higher levels of glycoside hydrolysis. and just shows you that the data from animal models cannot always be translated to humans. The real breakthrough with these types of drugs was to recognize that the glycosidic linkage has this issue of being metabolically labile and replace it by some mimic which is more stable. And remember, in a glycosidic linkage, we are linking a carbohydrate by this oxygen bond to some other unit, and it's that carbon-oxygen bond that's labile. And pharmaceutical companies shifted their attention to compounds which don't have that oxygen. So these are examples of compounds where we are calling them C glycosides instead of an O glycoside because there is no O. So now the carbohydrate is linked directly by a carbon-carbon bond to the rest of the molecule. And this bond will not be cleaved, of course, by a glycosidase since there's no oxygen here. And these C glycosides are now stable, so they contain a glucose at one end to provide binding interactions and recognition by the glucose co-transporter. And over here they have an aromatic group that again provides you binding, but they remain stable since they lack the O-glycoside. And at the moment there are 4 examples of this class from different companies that are approved. And in terms of their physicochemical properties, you can see that they are all suitable for oral absorption. So these are orally administered drugs. And you can see they're quite similar to each other in structure. They all contain a glucose unit, and this has then been linked by carbon-carbon bond to some aromatic moiety. And although it's not shown on this slide, they are highly selective for SGLT2 by thousands of folds compared to the SGLT1 that's present in multiple organs in the body. These drugs are not associated with weight gain, and in fact they promote some weight loss because there's reduced glucose circulating in the system. And for the same reason, they're not associated with hypoglycemia. Their effect is solely in the kidney, so there's no dependence on insulin secretion. The major side effect is that due to the presence of these drugs, glucose is not being reabsorbed efficiently in the kidney, and that means it's going to be present in large volume in the excreted urine. So with this drug, it causes glycosuria, which is the presence of glucose in the urine at a level of over 50 grams a day. And that in itself can cause side effects. So firstly, you have a high level of glucose in the urine that changes its osmotic pressure and leads to a more viscous urine. And this can cause tiredness, also dehydration. And furthermore, these high levels of glucose in the urine can then be used as food by microorganisms. So patients on these drugs are more susceptible to urinary tract infections for that reason. But nevertheless, these are quite successful drugs. They are the newest of the anti-diabetic drugs on the market, so fairly recently approved. So at this moment, they are still under patent, which means that although they are small molecule drugs, they are relatively expensive drugs compared to some of the older drugs that we have been looking at. And with that, we'll end this part of the lecture.